Welcome to today's Explainer. We're jumping right into a fundamental physiology topic today, and we're bringing some serious high-voltage energy to it. We are breaking down a fantastic study guide that maps out the literal biological mechanics of how substances actually get inside and outside of our cells. We're talking active versus passive transport.
The source material we're looking at actually kicks off with this incredibly enthusiastic greeting that I just have to pass right on to you. Hello, students! I hope you are all studying hard. The author promises to break down cellular transport so simply and so logically that you're going to easily score full marks on any test. So, let's figure out exactly when our cells get to kick back and relax and when they really have to do the heavy lifting.
Okay, here's our game plan for today. First, we'll hit the plasma membrane border, then cruise down passive transport, meet our protein helpers in facilitated diffusion, flow right into osmosis, hike up the active transport highway, and wrap it all up with the exam magic key. All right, Section 1. The plasma membrane border, the stage for cellular drama. Because, hey, before we can even talk about the traffic, we have to understand the borders, right?
What's really fascinating here is how this guide totally redefines the way we usually picture cells. The plasma membrane, it is absolutely not just some boring, static brick wall. It's a highly dynamic, selectively semi-permeable border control. It is constantly managing the hustle and bustle between the intracellular fluid inside your cell and the extracellular fluid outside. Think of it as the ultimate VIP bouncer. It decides exactly who gets in, who gets kicked out, and it manages two main biological highways to make it all happen.
Moving on to Section 2. Highway 1. Passive transport. The downhill road. This is our first major route, and it's basically the place where your cellular machinery finally gets to take a little breather. So the absolute rule of thumb for passive transport is that substances always, and I mean always, move down their concentration gradient. That means they're traveling from an area where they are super concentrated down to an area where there's less of them. Since this is just a completely natural phenomenon of physics, it's exactly like rolling a ball down a steep hill. It just happens automatically, and honestly, the best part for the cell? It requires absolutely zero precious ATP energy. The cell just sits back and lets nature do the work.
Now, this passive downhill highway isn't just one single boring road. The guide actually splits it into 3 distinct lanes, depending on who is driving. We've got simple diffusion, facilitated diffusion, and osmosis.
Lane number 1 is simple diffusion. This is basically a non-selective process where molecules just dissolve directly into the hydrophobic phospholipid bilayer of the membrane. Since they can dissolve right through, they cross the border easily without even asking for help. So who gets this VIP express pass? Mostly small non-polar molecules, think oxygen and carbon dioxide, or hydrophobic molecules. They literally just slide right through the lipid bilayer like ghosts passing through a solid wall.
Which brings us to Section 3. Protein helpers, facilitated diffusion, for the larger molecules. Now this raises a massive question. What happens when you aren't a tiny gas molecule? What if you're a large polar molecule, like a carb, or an amino acid, or maybe a charged ion? Well, you'd completely fail to dissolve in that hydrophobic lipid bilayer. You would just be stuck at the border.
Enter our protein helpers, who run the second lane, facilitated diffusion. Here, the crossing is helped out by specific membrane proteins that act as transporters. But, and this is key, don't forget where we are. We are still on the passive highway. These special lanes allow larger molecules to cross down their concentration gradient completely without spending a single drop of energy.
And that brings us to the 2 main flavors of these transport helpers. You've got carrier proteins on one side and channel proteins on the other. Both of them get the job done without any ATP, but their methods beautifully different.
Let's take a closer look at carrier proteins. These guys act incredibly similar to enzymes. They have these highly specific binding active sites for the exact molecule they want to transport. When that target molecule docks, it triggers a conformational change. The protein literally, physically changes shape. It flips to face the inside of the cell and just dumps the molecule right into the cytoplasm. But because they actually have to physically bind and flip, they exhibit what's called saturation kinetics, meaning if too many molecules try to cross at once, they hit a maximum ceiling. A perfect example of this is how your cells take up glucose using specialized GLUT transporters.
On the flip side, we have channel proteins. Instead of binding and flipping, channel proteins essentially just open up a hallway.
They create these hydrophilic pores directly through that fatty hydrophobic bilayer. Now, they are selectively permeable based on size and charge, and a lot of them are gated. If they open up because of an electrical change, they're voltage gated. If a chemical, like say a neurotransmitter, bumps into them to open the door, they're ligand-gated. These rapid-fire channels are basically the unsung heroes of your nervous system. They allow for the lightning-fast nerve impulse transmissions and muscle contractions that literally let you move and think.
All right, Section 4. Osmosis, the waterway, the third passive lane. We've officially arrived at the third and final lane of our passive highway, and this one is strictly reserved for a very special VIP, water.
Osmosis is the movement of a solvent, which is almost always water, across our semi-permeable membrane. The terminology here is absolutely crucial. Water moves from an area of lower solute concentration, meaning a dilute, hypotonic solution where water is highly concentrated, over to an area of higher solute concentration, a hypertonic solution where there's less pure water and way more dissolved stuff. Just think of water as always trying to dilute the most crowded party. It's going to keep moving across the membrane until the concentrations on both sides are completely equal.
So, why does this matter? Well, it is quite literally life or death for your cells. Osmosis regulates cellular water content, maintaining the absolute perfect volume. If you drop a cell into a super salty, hypertonic environment, water rushes out and the cell shrivels up like a raisin. Put it in pure water, it swells up and could literally burst like a water balloon. Osmosis is what keeps this delicate balance in check.
Moving into Section 5, Highway 2, Active Transport, the uphill battle. Okay, that covers the nice, relaxing, automatic downhill ride. But what if the cell desperately needs to pump a substance against the natural flow?
Imagine trying to push a giant, heavy boulder up a super steep hill. It is hard work, right? Pumping substances uphill against their concentration gradient, so from low concentration to high concentration, requires fighting physics. And the cell absolutely cannot do this for free. To cross this particular highway, the cell has to pay a toll, and that toll is spending its precious ATP energy.
Active transport uses highly specific membrane pumps to make this happen. Now, why would a cell spend its hard-earned ATP? Because it has to. It needs these pumps to forcefully remove toxic cellular waste against the gradient, to hoard essential nutrients, and ultimately to maintain homeostasis. Without spending this energy, our stable internal environment would literally just collapse.
And finally, Section 6, your exam magic key, the ultimate summary. We have covered a lot of biological miles today, so it is time to lock in the single most important takeaway from our source material.
The crucial point all comes down to this perfect contrast. Seriously, if you remember nothing else today, remember this. Passive transport is a downhill ride. It moves from high to low concentration, and it uses absolutely zero ATP. Active transport, on the other hand, is an uphill battle, forcing things from low to high concentration, and it demands ATP energy. Keep that distinction clear, and you will literally never confuse cellular transport again. To really make those exam answers pop, the source material offers this super actionable piece of advice. Always draw a neat diagram. Sketch out that lipid bilayer. Show it tiny molecules sneaking through via simple diffusion. Draw a bulky carrier protein flipping its shape, and a wide-open channel pore letting ions rush in. A visual representation proves to your grader that you truly grasp the concept. Oh, and a gentle reminder directly from our author, as you're preparing for those exams, do not forget to prioritize your own biology. Protect your sleep cycle. It's so incredibly important.
I'm going to leave you with a quick cliffhanger to test your new knowledge right now. Based on everything we've unpacked in today's explainer, is the uptake of glucose into a cell an example of simple diffusion or facilitated diffusion? Think back to our protein helpers. Study hard, stay focused, and keep exploring that amazing microworld inside of you.